Okay, so what are the stages of transcription? So we have three important stages for transcription given by IET. The first one would be initiation. The second is elongation. And the third one would be termination. So it's quite easy to remember. Initiation, elongation, and termination. And we'll be looking at the details a little bit later. But in summary, you can see here that initiation will simply what will happen simply will be the RNA would be attaching itself in the promoter sequence to start the transcription process. The elongation will produce the mRNA, and the termination will end the production of the mRNA sequence. So that's the whole overview of the transcription process. Let's now go into the details. Now, you can see here in this slide two important parts. One would be for the prokaryote, while the other one would be for the eukaryote. So take note of the very important difference. First of all, there are important characters in the prokaryotic transcription. First of all, or initiation, first of all, you have the presence of the RNA polymerase, and then you also have here the important portion, which would be the promoter sequence. Now, take note what happens in the prokaryotic initiation is that the sigma factor, which is a very important enzyme here, will be the one to recognize the promoter sequence in order to help the RNA polymerase find the promoter sequence and to start off the transcription. So you see here that the sigma factor will be recognizing two important portions, which would be the positive 35 region, and then you have here the negative 10 region over here. So it's very important for you to take note of that this would be the production or the start of transcription. Now, the consensus sequence wherein the sigma factor would be coming into play or will be attaching itself is called the Pribno box, which is a TAT-AAT, the TATAT, and you can see that over here, very important. So the Pribno box is important for the recognition by the sigma factor to attach to, and then the sigma factor is important so that the RNA polymerase will be able to recognize it and attach itself to the promoter sequence. And that's the start of the prokaryotic transcription. Now, comparing this with the eukaryotic transcription, you see here also the promoter sequence, but instead of a sigma factor, it has a number of initiation factors, transcription factors, and you have here the binding of several. One of these, we won't be mentioning all of this, but TF2D, TF2A, so all of this stuff are needed in order for RNA polymerase to recognize the promoter sequence and then attach to it in order to start the process of transcription. Now take note, if for prokaryotic transcription, the consensus sequence is called the Pribno box or TATAT. You have here for the eukaryotic transcription, you have here the consensus sequence called the TATA box, which would be the TAT-AAA. So that would be an 80-rich region. So take note of this one. So this one contains the transcription initiation complex for the eukaryote, while the prokaryote does not have any initiation complex. So that's for the process of initiation. So the next slide will just show us that the sigma factor for the prokaryotic transcription would need to recognize the negative 35 sequence in the Pribno box in order to attach to it, and then the RNA polymerase will be able to attach since the sigma factor is already there. For the eukaryotic transcription, you can see here the TATA box, which is important for all the transcription factors to attach themselves, and then the RNA polymerase will be able to attach and start transcription, which this is called the transcription initiation complex. Alright, now for the process of elongation, this is quite simple because it will be the same for both eukaryotic and also prokaryotic transcription. Here, the double-stranded DNA will simply be torn apart by the RNA polymerase, breaking down the hydrogen bonds between the base pair. So this will open it up in order for transcription to happen. So in this case, the DNA double strand, there will be what we call as a sense strand, and the sense strand will be also called as a coding strand or the non-template strand. This is the strand that will not be used for production of the mRNA. So that's why it's called a non-template strand. It is called the sense strand or coding strand. While on the other hand, the antisense strand, which would be the one that is being used, or the template strand, is non-coding and also called the template strand. So what happens here is that simply the RNAP, the RNAP will simply produce or use the nucleotide or the RNA nucleotides to produce your mRNA sequence here, or the mRNA. So there will be a growing strand here. So it will be added from the 5' to 3' direction in order to produce your mRNA. Okay, so this is what it looks like. So you have here the non-template strand, and then you have the template strand over here, and then here will be attaching or base pairing the RNA nucleotides with the DNA nucleotides of the template strand, eventually producing your mRNA. So that's the whole thing about the elongation process, where we're done with the initiation, we're done with the elongation. Now we proceed with the termination, wherein simply stated, the RNA production will now stop. There is a difference between the prokaryotic termination and the eukaryotic termination, precisely because for the prokaryotic termination, it is rho dependent, which means there is an added enzyme called the rho factor that will be needed in order to separate the RNA polymerase from the DNA strand, and this will end the production of the mRNA. So the rho factor recognizes the transcribed RNA, and then it will be able to read the termination sequence, and the terminator RNA sequence serves as the termination signal. So it will simply be there, or the RNA or rho factor will recognize this, and then finally kick out the RNA polymerase, thereby stopping the production of the mRNA. So it will be released when the rho factor catches up. Now take note, on the other hand, for the eukaryotic transcription, there is no such rho factor present. That is why it is rho independent. And then take note that the RNA polymerase will simply continue on transcribing the DNA sequence until it reaches the termination sequence that will contain the AAU, AAA, and then here it will be stopping. So it will continue on producing that sequence or the mRNA until it reaches the termination sequence, and then it will simply be dissociated or eventually dissociated, and that will stop the eukaryotic transcription. So the presence of this sequence will be very important for the termination in eukaryotic transcription. Okay, so let's just have some concept check. What is the directionality of the mRNA synthesis during transcription? The answer to this is 5' to 3' direction. So you see here that the RNA sequence will be added, or the nucleotides will be added from 5' to 3' direction. So this is the direction of synthesis. Another question here is the post-transcriptional processes in eukaryotes. So very important, right after transcription of the mRNA, at least for eukaryotic systems, you need to undergo post-transcriptional processes, and there are 3 important things here. There will be capping, there will be splicing, and finally the addition of a poly-A tail. So here in the post-transcriptional process, particularly splicing, there is the importance of the spliceosome, wherein you have the exons and the introns. The exons are actually sequence of the mRNA that will be coding for particular parts of the polypeptide, while the introns doesn't code for anything. So there is a need to remove the introns in order to produce your mature mRNA. So the spliceosome will remove the intron and then connect together all of the exons that are important. So again, the RNA processing here, this intron and intron are removed, and then the exon 1, the exon 2, the exon 3 are spliced together. There is also the addition of the 5' cap and the addition of the poly-A tail. So this would be all about the post-transcriptional processes. Okay, so here's the capping made up of the guanine nucleotide added to the 5' end, and then there will be several A's over here, 50 to 250 adenine nucleotides that will be added at the 3' end. Now, there's a question. After post-transcriptional process, where does the mature mRNA will be transported? The answer to that is it will be transported to the cytoplasm. So here you can see here that it will be translocated into the cytoplasm. Another question. What is the importance of capping and polyadenylation? The answer to that is The 5' cap protects the mRNA from degradation and helps ribosome binding during translation. And also, the poly A tail will be very important to protect it also from degradation and helps in the exportation of the mature mRNA to the cytoplasm. What is the difference between intron and exon? Well, the answer to that is the exons are nucleic acid coding sequence, while the introns are non-coding sequence present in the hnRNA, which would be the pre-mRNA. Okay, what is a large RNA protein complex that catalyzes the removal of introns from nuclear pre-mRNA? The answer to that is the spliceosome.